Welcome to this tutorial on cell and cell organelles, brought to you by Medical Biochemistry, Power Capsule series. This video pertains to the following CBME curriculum topic. Describe the molecular and functional organization of a cell and its subcellular components. You have been studying about cells since your school days, and therefore you have a pretty good idea about cells and cell organelles. In this chapter, I shall be emphasizing some key concepts regarding cells which you would find useful while pursuing medical biochemistry. The plasma membrane separates the cell contents from its surroundings and defines the periphery of the cell. It is composed of a lipid bilayer and protein molecules. Cholesterol too is an important component of the plasma membrane. The plasma membrane forms a thin, tough, flexible, hydrophobic barrier around the cell. The membrane is a barrier to the free passage of inorganic ions and most other charged or polar compounds. The individual lipids and proteins of the plasma membrane are not covalently linked, and therefore it is flexible and allows changes in the shape and size of the cell. The internal volume enclosed by the plasma membrane is the cytoplasm. It is composed of an aqueous solution, the cytosol. Cytosol is therefore the fluid portion of this cytoplasm. A variety of particles with specific functions remain suspended in cytosol. Suspended in the cytosol are special structural components which we know as the organelles. Individual organelles are well organized and well demarcated by membrane. The entire substance of the cell bound by plasma membrane is known as protoplasm. Cytoplasm refers to only protoplasm outside the nucleus. The plasma membrane has different macromolecules embedded in it, and these have various functions. These molecules may include receptors, signal molecules, ion channels, transport proteins, enzymes, etc. Transport proteins in the plasma membrane allow the passage of certain ions and molecules. Receptor proteins transmit signals into the cell. Membrane enzymes, such as alkaline phosphatase and 5' nucleotidase, participate in some reaction pathways. Ions and molecules are transported by the channels and transporters through passive or active transport mechanism. Plasma membrane molecules have significant applied importance, as abnormalities in their structure and function have been linked with many disorders. We shall present a detailed discussion on plasma membrane, the embedded molecules and their function, in a separate tutorial. Nucleus is the controlling center of a cell, because it contains DNA that regulates all functions of the cell, including growth and metabolism. Chromatin is the DNA organized with proteins. Nucleus contains an area visible as lightly shaded, where RNA processing and ribosome synthesis take place. This is the nucleolus. The nucleoplasm, which is the protoplasm of the nucleus, is bounded by a double membrane, nuclear envelope, which has pores on its inner membrane. Outer membrane is continuous with endoplasmic reticulum. From the viewpoint of genomics, nucleus is a membrane-enclosed organelle within a cell that contains the chromosomes which encode genetic material. The endoplasmic reticulum is a complex network of interconnecting membrane channels that are continuous with the nuclear membrane and plasma membrane. In most cell types, it can be considered as the largest organelle. Major steps of protein synthesis take place in endoplasmic reticulum. Detoxification of xenobiotics by cytochrome P450 system takes place in endoplasmic reticulum. Ribosomes, the molecules responsible for synthesizing protein, are attached on the rough endoplasmic reticulum, thereby giving these the rough appearance. The endoplasmic reticulum is analogous to a busy industrial factory complex. The endoplasmic reticulum is a large dynamic structure that has roles in protein synthesis, protein transport, protein folding, steroid synthesis, lipid metabolism, carbohydrate metabolism, and calcium storage. Distinct domains, such as tubules, sheets, and nuclear envelope, perform the diverse functions of endoplasmic reticulum. Every student of biology knows that mitochondria are powerhouses of the cell. Mitochondria have their own specific DNA known as mitochondrial DNA. Mitochondria have two membranes, The inner membrane is convoluted into folds called cristae. The inner membrane contains ATP synthase and other components of the electron transport chain, all of which are involved in ATP synthesis. The inner membrane encloses the fluid matrix, which contains various enzymes that catalyze metabolic reactions. Mitochondria have a good number of important functions, apart from its well-known function of ATP generation. Mitochondrion is also the organelle where important metabolic reactions related to carbohydrates, lipids, proteins and nucleotides take place. Golgi apparatus is an important cell part in connection with protein synthesis and protein maturation. Golgi apparatus was first described by Camillo Golgi. Golgi apparatus is a network of flat and smooth membranes and vesicles. It's the organelle where proteins, particularly glycoproteins, are given their final form. Golgi apparatus is composed of three zones: cis, medial and transcisternae. Protein sorting, packaging and secretion are the main functions of Golgi apparatus. Lysosomes are tiny organelles containing various enzymes encapsulated within. The purpose of lysosome is to hydrolyze various macromolecules, thereby disintegrating them. Enzyme-enabled hydrolysis by lysosomal enzymes is required for digesting foreign substances that have entered into cells. Lysosomes may also digest components of the cell itself by hydrolyzing various constituent macromolecules in a process known as autophagy. In micro-autophagy, cytoplasmic proteins meant to be degraded are taken up by direct invagination by lysosomal membranes. In chaperone-mediated autophagy, proteins are flagged with markers, which are then selectively taken up by lysosomes and then degraded. In macro-autophagy, unwanted substances are taken up by subcellular structures, called autophagisms, and then delivered to lysosomes for breakdown. Lysosomal enzymes are responsible for post-mortem autolysis. These enzymes also have a role in gout cancer metastasis and silicosis. Inclusion cell disease and lysosomal storage diseases are caused due to suboptimal or grossly reduced functioning of various lysosomal enzymes. In short, lysosomes break down excess or worn-out cell parts and have a role in destroying invading viruses and bacteria. They are the suicide bags of the cell because of their role in programmed cell death or apoptosis. The centrosome is a non-membrane-bound organelle and is composed of two centrioles surrounded by the pericentriolar matrix. Centrioles are microtubule rings. Centrosome facilitates spindle formation in cell division and organizes the spindle fibers. One of the main roles of centrosome is to organize microtubules as well as pull chromatids apart. During cell division, centrosome, being the major microtubule organizing center, also stabilizes the structure of the cell because microtubules are components of cytoskeleton. Centrosomes divide prior to cell division. Centrioles are composed of microtubules arranged in barrel-like structure. Centrosome has a role in maintaining the chromosome number during mitosis. It controls cell polarity. It also stimulates cell membrane shape changes during phagocytosis. Dysfunctions in centrosome can cause cancer of liver, colon, and breast. Peroxisomes are organelles which are relatively more abundant in leukocytes and platelets. Peroxisomes contain the enzymes peroxidase and catalase. These enzymes can destroy unwanted peroxides and free radicals, which have deleterious effects. Peroxisome is involved in biosynthesis of ether phospholipid, plasmalogens, cholesterol and bile acids, some catabolism of purines, polyamides, eicosanoids, prostaglandins, very long-chain fatty acids, and glyoxylate clearing takes place in peroxisomes. Peroxisomes may have a role in inflammation, ischemia, reperfusion, and diabetes. They have a key role in both production and scavenging of reactive oxygen species. Peroxisomes generate significant amounts of hydrogen peroxide and reactive oxygen species. However, they also contain multiple antioxidant enzymes, such as catalase, superoxide dismutase, glutathione peroxidase, epoxide hydrolase, peroxyredoxin 1, all of which can regulate reactive oxygen species. Peroxisoms can proliferate or be degraded in response to nutritional and extracellular environment stimuli by peroxisome proliferators, which include various chemicals, toxins, certain lipids, etc. Hence, if more peroxisomes are formed due to influence of peroxisome proliferators, then there is imbalance in formation and scavenging of reactive oxygen species, leading to oxidative stress. This mechanism may have a role in cancergenesis. For the same reason, proper functioning by peroxisomes may have a protective role so far as cancer and neurodegenerative diseases are concerned. Adrenoleukodystrophy, Zellweger syndrome and primary hyperoxaluria are three noteworthy disorders in relation to defective peroxisomal function. There are of course other diseases in the spectrum which haven't been mentioned at this stage. Peroxisomes have been discovered not so long ago, and we have just begun to understand their immense role in health and disease. The cytoskeleton is a structure that helps cells maintain their shape and internal organization. It is an interconnected network of filamentous polymers and regulatory proteins and is dynamic and adaptive. The architecture of the cytoskeleton controls the physical properties of the cell. The three main cytoskeletal polymers are actin filaments or microfilaments, microtubules, and intermediate filament group. Microtubules are composed of tubulin and actin filaments are composed from actin. Intermediate filaments are composed from different types of subunit proteins, which vary from cell to cell. Microtubules are anchored in centrosome and radiate out from the centrosome. Cytoskeleton spatially arranges the contents of the cell, arranges and maintains the integrity of intracellular compartments, and is important for positioning of organelles. It connects the cell to the external environment. It generates coordinated forces that enables the cell to move and change shape. It provides mechanical support and maintains cell shape. The microtubules along with the centrosome has important role in cell division. Therefore the cytoskeleton provides structure and organization, resists and transmits stresses, drives shape change and movement, and provides mechanical support that enables cells to carry out essential functions during cell division. We now have a picture of a generalized cell. In reality, cells come in a wide range of varieties. The exact shape, size, and organelle content depend on the nature of the cell and its biological purpose. The organelles can be taken out of the cell after breaking the cell membrane using solvents or mechanical disruption using special techniques and equipment. This is the process of homogenization. Since the organelles have different densities, they can be conveniently separated by differential centrifugation. Some organelles have special chemical identifiers, known as marker enzymes, through which the organelles can be identified after centrifugal separation. This knowledge is particularly required by those who are involved in experimentation and technical work related to biology and biotechnology. Cell biology, being the study of cell structure and function, enables us to have a detailed understanding of the tissues and of our bodies that cells compose. When we study biochemical mechanisms and reactions, we are essentially delving into the study of cell metabolism.
Thanks for watching this video. Our next tutorial in the Medical Biochemistry Power Capsule series is titled, The Plasma Membrane and Its Functions. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids, and for purpose of rapid revision prior to exams. Please don't forget to like, subscribe and share.